All right, good evening. My name is Hannah Antosh, and we will be covering Mendelian inheritance. Before we begin the information portion of this presentation, I wanted to begin with some applicable introductions to concurrent credit students. My first option, and one of my favorite options, is to have students create a poster of any one law and then explaining it in front of the class. Anytime that I can combine art and science, I like to do so. I find that it helps students retain information better. By doing this, not only are we unlocking the creativity of students, but also we are making them learn. Super fun stuff. The second option, and one of my other favorites, are coin flip creatures. I mentioned this later in the slideshow. This is a great way to introduce the law of independent assortment. That is the law that alleles are inherited independently from each parent, right? Completely at random. This is a great way to do that. I like to give students a list of traits that their creature can have, hand them a coin, and let them flip away, right? They get to, based on the outcome of their coin flips, that determines what traits their creature may have, whether they be dominant or recessive. And that's a great way to show students how these alleles are inherited separately, right? The coin flips are not related to each other. Just because you flipped heads on one coin flip does not mean that you will flip heads on the second coin flip. My third option is I do love having students create presentations, and I do love having students research scientists of the past. I would love to have students create a presentation on Gregor Mendel, explaining who he is, or who he was, where he came from, and how he came to be known as the father of modern genetics. Some of the objectives of this presentation are to summarize the life, the faith, and the scientific contributions of Gregor Mendel, to describe Mendel's laws of dominance, to describe Mendel's law of segregation, and finally, to describe Mendel's law of independent assortment. Why are Mendelian genetics important to understand? First and foremost, I find that researching and learning more and understanding where our science came from, who it came from, gives us a greater appreciation of the progress that we have made. By understanding who Gregor Mendel was and what he had to do to understand genetics today, it creates a great understanding and a great appreciation of how far we have come. Mendelian inheritance allows us to understand how genes and traits are passed on in a fairly straightforward manner. These inheritance patterns are very important in understanding dominant and recessive traits, dominant and recessive genes. By knowing which genes are dominant and recessive, geneticists can help to identify couples who may pass on genetic disorders through their alleles. Some diseases, such as cystic fibrosis, Tay-Sachs, and sickle cell anemia, are all known as autosomal recessive disorders, meaning that each parent must carry a copy of the recessive gene for that disease in order for their offspring to inherit it. Before we dive into the laws, I want to take a moment to talk about Gregor Mendel. He was born on July 20th in 1822 in Heisendorf in Silesia in the Austrian Empire. He died on January 6th, 1884 in Brunn, Austria-Hungary. Gregor Mendel was a fairly academic kid from the start. He joined the Augustinian order, where he was able to flourish academically due to the time he had to devote to his studies. During his time in the order, he experimented extensively with pea plants, during which time he discovered his laws of inheritance. He is now known as the father of modern genetics. Those laws that we are going to talk about in order are, number 1, the law of dominance, number 2, the law of segregation, and number 3, the law of independent assortment. The law of dominance asserts that one allele will be expressed more strongly than another one. Mendel came to this discovery while he was experimenting with his pea plants. He noted that when he crossed peas that produced wrinkled seeds and plants that produced smooth seeds, when he crossed those two, all of the offspring had smooth seeds. The prevailing genetic theory at the time were convinced that the offspring tended to have blended traits from both parents, which is the case in some traits, but not this one. Mendel disproved this particular theory, in simple inheritance patterns of course, by demonstrating that the wrinkled seed crossed with the smooth seed, one did not get a semi-wrinkled seed in the next generation, but rather a smooth seed, which went against what people assumed would happen. Mendel's discovery of dominant and recessive traits did not just extend to the outward or the phenotypical appearance. He also discovered that genotypes, the alleles that the organism possessed, also played a role. In fact, genotypes lead to phenotypes. In the second generation, Mendel recrossed those two offspring. When he crossed those two offspring together, those two smooth seed-bearing plants, when he crossed them, they did not produce exclusively smooth seed-bearing plants. Instead, though the offspring of that generation produced a variety of wrinkled and smooth seeds. Thus, Mendel discovered that organisms could be heterozygous, which means they bear dominant and recessive alleles, or they could be homozygous, bearing dominant or recessive alleles. This is very important. It's so important, in fact, it leads to the next law, the law of segregation. The law of segregation asserts that each parent contributes one allele per gene to the genetic makeup of the offspring. Unfortunately, I do like to take this time to remind us that there are times where parents accidentally donate too much. In aneuploidy, such as trisomy, their 3 chromosomes make it into the offspring. In Down syndrome, this is typically trisomy 21, where there are 3 copies of the 21st chromosome. This can occur due to mishaps during meiosis when the chromosomes do not separate correctly. Now, if we refer back to our second generation cross from the last slide, assuming that the wrinkled parent has the same genotype, little w, and the smooth parent has a genotype, big W, when we cross those two, all of their offspring must have the genotype, big W, little w, because each parent can only give one allele. Only one, no more. When the offspring are crossed back to each other, right, that big W, little w, when 2 of those are crossed together, their offspring have 3 possible genotypic combinations. Big W, big W, little w, or little w. Remember that big W means that those plants are going to be giving us smooth seeds. With big W, little w, because we have that dominant W allele, we're still going to be getting smooth seeds, even though there is a recessive allele. Now, the only time that we will be getting recessive phenotypes, remember, when the physical manifestation of that recessive allele is when the offspring have 2 recessive alleles, right? When they have this little w, this is the only time that we will be getting any kind of recessive genes. If you're super nerdy and you would like to do a little bit of further study, I find that the royal family's history with hemophilia or the Habsburg chin are incredibly interesting case studies of when we can see directly not only the impacts of inbreeding, thank you Habsburgs, but also dominant and recessive traits. It's a very, very straightforward case most of the time. This is going to lead us to our next law, the law of independent assortment. The law of independent assortment says that alleles of a gene are inherited independently of each other, totally at random. Think back to our coin flip creature. If we were for a moment to imagine that we are tracking two different genes, the shape of the seed pod, for example, and the color of the flower, that's what we're going to track in our imagination for just a minute. Say that the genotypes for the seeds are the same as last time, big W, little w, as are the phenotypes, right? Wrinkled and smooth. Now, if we want to add in flower color, this is where it gets fun. A purple flower, assuming that the purple flower is dominant to the white flower. In this example, each pea plant can either have big W, big W, big W, little w, or little w, little w. Those are the only genotypes that they can have in regards to whether their seeds are smooth or round. Now, if we move on to flower color, we're going to use P. Big P is going to stand for purple, and little p will stand for white. When we have big P, right, we're going to have purple flowers. When we have big P, we'll also have purple flowers. But when we have little p, we will get white flowers. When we combine these two genes and we track them simultaneously, our genotypic options are big W, big P. And in this one, the flower is going to have smooth seeds and purple flowers. We can also get, again, smooth seeds, two big W's, and purple flowers because we have the big P. We can also have big W, little p, where we have, again, smooth seeds, but now we have white flowers. We can also get more smooth seeds and purple flowers with big W, big P. Or we can go for a true heterozygote and get big W, big P. Again, we're going to have wrinkled seeds and purple flowers. We can have big W, which is again smooth seeds, but now we have white flowers. We can also have little w, big P, in which we get wrinkled seeds and purple flowers, wrinkled seeds again, and purple flowers again, or finally, we can have wrinkled seeds and white flowers. Each one of these alleles was inherited independently of the others, right? This little p, this recessive allele, was inherited independent. It didn't depend on another recessive allele or another recessive allele. It came on its own. And that is the law of independent assortment. If we wanted to do a little bit of further information into inheritance, because I think it's super fun, Mendelian inheritance patterns are an amazing tool. But there are some traits that do not necessarily obey these laws. We call these non-Mendelian inheritance patterns. These traits can be passed through, for a few examples, codominance, incomplete dominance, or even polygenic traits. Below are examples of these. ABO blood types are an example of codominance because neither the A nor the B are completely dominant over the other one. Neither the A antigens or the B antigens are not dominant over the other one. We could also have skin color. Skin color is a great example, as is hair color, and eye color of traits that don't necessarily obey Mendelian's inheritance patterns. These are in fact examples of polygenic traits. Other examples of polygenic traits are genetic conditions that can create cancer, diabetes, and Alzheimer's, among other conditions. What that means is that there has to be multiple traits in order for these things to occur. Skin color is a great example because it takes multiple genes in order to determine a person's skin color. It is not necessarily a straightforward answer. Thank you so much for your time, and this was Mendelian Inheritance by me, Hannah Antosh.